Now we can move on with Susana Fuentes Vélez from the Department of Electronics and Telecommunications of Politecnico di Torino. She will give us a speech entitled Impedance-based microfluidic devices for personalized medicine applications. Susana achieved her Bachelor of Science and Master of Science degrees in Biomedical Engineering thanks to a double degree program between the Universidad EIA in Medellin, in Colombia, and the Politecnico di Torino in 2018. She developed her master's thesis in association with our research and development laboratory of the company Eltec SPA. Since October 2018, she started working in the MINES research group under the supervision of Professor Danilo De Marchi. On November 2019, she started a PhD program in Electrical Electronics and Communications Engineering, collaborating with the Molecular Biotechnology Center at the Università degli Studi di Torino and the company Eltec SPA. Her main research interest is found where biotechnology meets electronics. Well, Susana, it is your turn. Thank
you. Okay, thank you very much. Thank you to the IEEE Politecnico Student Branch for the invitation. I'm very happy and very glad to be here and have the opportunity to present a little bit of what I've been working on lately. So, as I was well introduced, I belong to the MINES research group from Politecnico di Torino. We collaborate with the Molecular Biotechnology Center and the company Eltec. So, as the title you can see, I will be talking about microfluidic devices for personalized medicine applications that are all based on impedance measurements. I'll try to be very brief because it's late. We all might be a little bit tired, and I'll try to focus on the applications. I will talk very little about theory and focus on applications and results. Of course, I'll give a brief introduction, and then we'll pass to our first application that I've called the 2D, two-dimensional application, because it's about measuring cell impedance. Here we'll be talking about cell cultures, how impedance can be used for cell monitoring, cytotoxicity tests, and also the results we're all most happy about, that it's drug-resistant characterization. Then, following the trend in biotechnology of the 3D cell cultures, we will move to the application I've been working on in the last days, that it's about translating the system that we validated in 2 dimensions to 3 dimensions. And then we'll conclude. So let's get started. So I work by bringing together microelectronics and biotechnology. I focus mainly on impedance measurements, and there are several advantages of using this kind of measurements in biotechnology. As you can see in the graph, it's a label-free technique. Also, it's quantitative. It's relatively low cost because it doesn't need a lot of reagents, and it's a real-time technique. It allows real-time monitoring. And what's most interesting, that it's not invasive and non-destructive for the biological samples. So I'll present our first device that is about impedance measurement to perform long-term cell culture monitoring, and then move to the 3D system. So a little bit of a few words on what it's measuring cell impedance. In simple words, impedance can be defined as the overall opposition of a system or circuit to a current when a voltage is applied. It can also be defined as a complex ratio between voltage and current, and it's made of real and an imaginary part. Therefore, it can be defined, as you can see in the screen, in terms of magnitude and phase. The potential of measuring impedance is that we can obtain information about the cell cytoplasm, the cell membrane, cell adhesion, cell proliferation, and other phenomena that we can relate to physiological and pathological conditions. The methodology that we're following is called ECIS, that stands for Electric Cell-Substrate Impedance Sensing, and relies on the single shell model of the cell. In this model, as I said, the cytoplasm is it's modeled as as a sphere of homogeneous phase, and the membrane as a thin shell. The great potential of measuring cell impedance of using impedance in cell culture monitoring is that it allows us to perform continuous monitoring. This represents a great advantage compared to traditional or conventional endpoint assays, in which we're only able to perform measurement at defined time points, and in most cases are destructive for the cell culture, forbidding, of course, subsequent analysis or subsequent assays. So how is it done? What do we do? As you can see in the graph, with electrodes at the bottom of the culture plate, the cell membranes of adherent cells interfere with the current flow, causing therefore an increase in impedance upon cell proliferation and adhesion. As you can see in the lower part of the screen, cell impedance spectrum can be defined in different ranges. At low frequencies, there's a cell membrane polarization phenomena. So we can get information about cell size and also with the proper setup about the charge transfer at the interface. At intermediate frequencies, cell membrane polarization phenomena is reduced, so we can have information about cell membranes. And at higher frequencies, the current can go through the membrane, so polarization phenomena is basically it's minimally reduced, basically neglectable, and we can have information about what's inside the cell cytoplasm, organelles, and even nucleus. So here's the system we worked on when we came out with this system that is based on a traditional, let's say, standard dimensions 48-well plate. So we're able to perform multiplex simultaneously analysis on 48 wells, and they have interdigitated gold electrodes at the bottom. So what do we got here in the graph? That it's a typical impedance-based cell monitoring curve. We have an x-axis, we have the time in hours, and the y-axis we have the normalized impedance. As we can see, we're able to see all the stages of adherent cells in vitro. So we have a first stage that it's a log phase that corresponds to adaptation time and addition. Then we have the exponential population increase. Then we have a plateau phase due to contact inhibition, and then cell detachment and death. The jump that's seen over here corresponds to a culture medium change. So actually validated that through impedance, we're able to perform long-term cell culture monitoring. We also perform addition tests. Through impedance, we're able to assess the addition of cells to different substrates. And we also perform a drug cytotoxicity test, as the one I present here. For this purpose, we were working with two different cell phenotypes. HT29 is a colon cancer cell line, and we were working with a wild type phenotype and a drug-resistant phenotype. So we tested different concentrations of a drug, in this case doxorubicin, and we're able to monitor what happens to the cells upon drug exposure. The red curves you see correspond to drug-resistant cells, and black ones to drug-sensitive ones, wild-type ones. So we can observe that upon drug exposure at, let's say, low, medium concentrations, drug-resistant cells keep on living, keep on proliferating, while drug-sensitive ones die. The nice thing about this is that we can, let's say, obtain real-time, understand what happens in real time to our cell culture. A very nice thing also is that through this data, we can obtain a regular, let's say, traditional drug response graph. Also working with these two cell lines, this was the most exciting result we obtained from this project, and it's that just by means of long-term culture monitoring. So this is no cytotoxicity assay, there's no reagent adding at any point. Just by monitoring the cell culture, we found out that differences in the impedance monitoring curves of drug-resistant and drug-sensitive phenotypes. As you can see here again, we have the red curve that represents the drug-resistant phenotypes, and the black curve that represents a drug-sensitive one. As you can see, they differ. And in the red curve, drug-resistant phenotypes, there's a significantly shorter plateau, suggesting a higher migratory tendency. We performed a gene expression analysis, and we found an enhanced mesenchymal gene expression that suggests an EMT process. EMT stands for epithelial to mesenchymal transition, that it's a process that affects migratory and adhesive characteristics of cells. The difference in the curves are also explained by the differences in the cell membrane composition of the drug-resistant cells, and also to the higher level of spreading that drug-resistant cells present. As you can see here, wild type tend to be more round, while these ones, drug-resistant ones, tend to spread more. So this was a very nice finding, because it means that we're able to characterize drug-resistant cells just by monitoring them, just by means of monitoring the cell culture through impedance. So this is the, let's say, the results that we recently published. Then, following the trend of biotechnology that says traditional cell cultures do not mimic the in vivo conditions, we should move to a 3D, more tissue-like reality. The idea is to come out with a system the same way as the one that we developed for traditional cell cultures, but made for 3D cell cultures, organoids, tissues, or whatever we want to measure in a 3D reality, let's say. The application is not so straightforward, though, because having a 3D construct, biological construct, means that static fluidic conditions are no longer suitable. So there's a need of a microfluidic integration, a perfusion system. All the physical parameters change, so of course we have to assess again all the measuring parameters, and in order to understand what we're measuring through impedance, since you saw that the impedance spectrum is wide, we need to integrate other physical or biochemical sensors. So it's more like an integrated platform. For this purpose, we started with the, let's say, stepwise process, traditional design process and simulation and fabrication, and then validation. For the design, we follow a translational approach, trying to follow their requests from the people, doctors and people from biotech. Also, because there's microfluidics in here, there was a need of simulation, because simulation is always nice in microfluidics. Then the first validation consisted on a flow and leakage test, let's say routine test in fluidics. And now here we're at this stage that it's biological testing, where defining the protocols, performing multi-frequency analysis, optimizing the measuring board, and so on. It's still, let's say, looking messy right now. So we can conclude that our system, our custom-made ECIS system, it's able to perform real-time monitoring of cells, as well as other tests such as addition tests and drug screening. Most interesting result is that we're able to perform drug-resistant characterization based on impedance. And from the 3D project, we can now conclude, even if it's a still ongoing project, we can said that the development of this kind of devices represents a step towards the goal of a point-of-care device. let's say that the application of all these devices are towards the use of cell-derived patients, in which by using the patient cells, we can define the actual therapy that is suitable for them. That's the idea of all the cytotoxicity tests and so on. So I hope I've been brief and also clear enough. Thank you very much for your attention, and I'm open for questions.
Thank you, Susana. Now it is the turn of your question and answer session. Well, we have a couple of questions. The first one, okay. Do you predict an application of this technique in wearable devices?
Wow, that's a very nice question. I've never thought about it. Wearable devices? I don't think so. This is more like a bench kind of equipment. Let's say going to downscale in order to have something that's wearable. Let's say it's suitable for lab applications in which still some protocols are needed. Let's say that impedance measurement, though, can be used for wearable applications. Like the concept of impedance, it's already used. You know, those their system that measure your body fat, like people from fitness, all those kind of measurements are based on impedance. But let's say the kind of study I'm interested about, that it's more about assessing drug efficiency and that no, I don't think it can be made wearable. But yeah, impedance, definitely. Through impedance, there are many kind of measurements that can be done.
Okay, thank you, Susana, for your very detailed answer. And then we have another question. What is the duration of a measurement to obtain a reliable reading?
Okay, so it depends a lot on the kind of culture, on the kind of cells you're using. So there's not a unique answer for that question. The nice thing about this kind of measurement is that it's upon to you the time of that you want to perform the reading. Let's say if you have neurons, or if you have colon cancer cells, or if you have cardiac cells, they have cell cycles in vitro that are different one from the other. In some cases, there's interested in performing short-term measurements, and by short-term, I mean a couple hours, 12, 24 hours. In other cases, it's more suitable 48, 72 hours. So it depends on the cell cycle and the cells you're using, of course. And also depends on what you wanna get from the measurement. If you wanna see the cell cycle, then a longer measurement is needed. If you wanna see addition, perhaps after 5-7 hours, or it depends on the cell type, you can already have your result. Like if you just wanna assess if the cells adhere or not through impedance measurement, then you just need the first part. Let me show you here. Perhaps it's clear. You just need the first part of the curve. Let's say if you want to evaluate addition. If you want to evaluate the cell cycle, then you need a longer measurement. So yeah, it's flexible. It's upon the protocol what you are interested in.
Okay, that's all. Thank you, Susana, for your very interesting talk again.